we're going to talk about the adaptive immune system today. The adaptive immune system is super cool. I think that it's interesting. Sorry, it's okay. It's really interesting to think about that our own cells are so complex and they're so specialized that they're so effective at what they do. You know, for the amount of people that have autoimmune disease, yes, people do have autoimmune and it is common, more common than we would like, but really in the scheme of things, actually we don't have that many autoimmune diseases. Oh, we're done with our note cards today, right? We're done, yeah.
I think you said we had one more now.
Entropius, we did pinworms, right? Huh?
There was a note card on the slide.
Oh, there was?
Or maybe that was last week.
Oh, yeah, I don't think there should have been. It was Enterobius, I think we're done, right? Pinworms was our last one.
Oh, yeah, we did do that.
Yeah,
We did do
yeah,
that.
okay, then I think we're done. Okay, so we're gonna talk about the adaptive immune system. So without the adaptive immune system, we would not survive. And it's incredibly powerful, it's rapid, it's the reason why we're not sick all the time, why if one person gets a cold, we don't all get it. You know, even if you might get a cold when somebody else has a cold, or you might get the flu when somebody else has the flu, you might get COVID when somebody else has COVID, why doesn't a whole class full of 30 people have COVID if one person has it? It's because of our adaptive immune system. Our adaptive immune system is so powerful. And you know, it's incredible to me also how powerful it is, but also how smart it is that our adaptive immune system is not also targeting our own cells so much so that everybody has autoimmunity. So it's smart, it's powerful, and it's the reason we're alive. So very cool. We're gonna talk a ton about antibodies today. So this is what an antibody is. You know, you go out to talk to people on the street, and everybody's heard of the word antibody, probably, but does anybody really know what an antibody is? No, probably no one. And so antibodies are very cool. I think everybody should know what they are, especially because they are talked about so frequently, and because they're one of the most important parts of our immune system. So we're going to talk about what antibodies are today and how they're made and what they do, and then we're going to talk about the whole adaptive branch of our immune system. So we talked about the first line of defense, we talked about the second line of defense, and now here we are in the third line of defense, and we're going to talk about B cells, T cells, and antibodies, our main topics today. But just so you know, just please ask a lot of questions if you're confused. I'll try my best to answer them from my immunology background. But I do love the immune system. It interests me a lot. I love to learn about it. And yeah, just keep asking questions because I think that it will help all of the students to understand it. Because each cell has its own really unique characteristics, and each cell has its own jobs. And those jobs are complex and they're powerful. So let's try and figure out what those roles are and make sure everybody is on the same page and caught up. Okay, so here we are in the adaptive. So this is our third line of defense. So innate is inborn, ready to go. It's less powerful, less effective. It's non-specific. It's broadly tries to kill stuff and get rid of stuff. You know, if you think about what the powers are, what the roles are of our innate cells, like if you look at these roles, like how powerful are these at killing bacteria? Yes, histamine and heparin they will call in more cells, they'll turn up the heat, but how powerful is that at get rid of a bacteria or a virus? And then these ones that phagocytose, yes, phagocytosis will kill the bacteria, but one at a time. So like how powerful is this system? It's not really that powerful. But I mean, we have to have it, yes, but it's not as powerful as the adaptive immune system. You'll see what the power of the adaptive immune system today. And the biggest and coolest thing about the adaptive immune system that I think is the specificity of every single thing we've ever been exposed to and that we don't get it. You know, how many things has my kid come home with and she doesn't get it, I don't get it. How many things are on every single thing we do and touch and we don't get it? It's because our immune system is so powerful and because it's specific to every single thing that we've ever been exposed to. So we'll talk about specificity today and how that happens. And so it's long term, it's cells that can be stored forever. We get a polio virus. Remind me if you guys can. We might forget because we can talk about it on Thursday as well. But we're going to talk about vaccines again because I want to bring that up because we get the polio vaccine, which probably most of us have had, and so we can go and work with people with polio. We can go work with people with polio and we will never get it. And because we had a shot when we were 1 month old or 1 day old or however, whenever we got it. And that's how long term we're talking about. It's slow to respond and we'll figure out why it's so slow. Why do we have these checks and balances? Why does it need to be activated? What is going on in the background? Why do we get sick for 5 to 7 days or 10 days? What is going on in that time for the adaptive immune system to kick in? And yeah, it's really powerful and the key is that second exposure. Yeah, go ahead.
Just kind of curious about when you brought up polio.
Yeah.
If we're vaccinated against polio, we can go work with people who have polio and not get it.
Yeah.
Is there any possible way to get polio after you're vaccinated, or is it just?
Well, that's a good question, and it kind of is a good review question too, because you know some vaccines you need boosters for, and those vaccines that you need boosters for, sometimes it's because the virus or bacteria, mostly viruses, because they're, well, I don't even want to say mostly viruses, because you do need a lot of boosters for bacterial vaccines as well, but because of how potent, so how virulent is that virus or bacteria, but also our B cells will die over time. So polio generally is a lifetime and you don't need a booster for that one. But also what calls to mind what is a good review is RNA versus DNA viruses and how often they mutate. So we do need to get it's basically like a booster every time that we get a new flu vaccine. But we get boosters for like varicella zoster virus, for the chickenpox, for shingles, because when we had the chickenpox, we might not have had enough of a viral load to store enough B cells to fight for the next time we have it. So there's a few things that go into that. Yeah, good question, yeah.
And there's some vaccines that are supposed to give you a lifetime immunity, but when you go into the nursing program, they will make you check and see for sure if you
do your titer.
have immunity or not. And if you don't, then you get to do it again.
Yeah, yeah. My daughter had to do it again, made me real mad, but yeah, sometimes you do. And especially you guys are working with so many patients, you know, you'll be seeing a ton of patients who have all kinds of things. So it's a good idea to just, you know, especially you don't want to be bringing it home to your kids. You know, if you have a brand new baby at home, that brand new baby doesn't have the same strong immune system that you have. So you could bring it home and give it to your child, even though you're, you know, you are not sick. So yes, washing hands, washing clothes, don't walk with your shoes in the house, all that kind of stuff. But also, the more that you're vaccinated, you protect your kids who might not be able to be vaccinated right away as well. Okay, so adaptive immunity, B cells, T cells, we're going to talk about those today. We're going to talk about what antibodies are and where they come from. Okay, so here's our adaptive cells. This is the ones that we're going to learn about today. Let's go through them. So what is a T cell? So a T cell, it's born in the bone marrow. Say that 5 times fast. It matures in the thymus. So when we say a T cell, when we kind of say T cell or when we say B cell, we're talking about like progenitor cells. So they will further differentiate into a T cell line. So like one T cell will actually change into different types of T cells. It's a stem cell for other T cells. So it will give birth to these other types of T cells. So there's many types of T cells. We're gonna talk about 3. We won't really talk about Tregs that much. We're gonna talk about 2 main ones. So T cells travel throughout the immune system. They reside in the thymus still till death. They in the lymph nodes and in all of our tissues and the lymphatic system. They run around in the lymphatic system. T cells especially are essential to immunity because we're going to talk about CD4 T cells when we talked about HIV and bring those back into play. Yeah.
Sorry,
No, please ask as many questions as you can. Yes.
I'm just kind of confused on like trying to picture a lymph node in their body. Like where are they attached to, or
Yeah. Let me
I
pull
know they're
that up.
kind of spaced out over the body.
No, I think that is good to bring up. Let me pull that slide up again. Because lymph nodes are everywhere, and lymph nodes are actually really cool and complex. If you ever do end up taking immunology, they'll show you, because lymph nodes have different areas of the lymph node where cells are stored and where they mature, and like it's this crazy, really complex little organ. Where was it? It was under innate.
I think it's 23.
Oh yeah. So here. So there's lymph nodes all over the body. We have, I forget how many, hundreds? I forget exactly how many lymph nodes we have. But there are lymph nodes everywhere. And these are just really cool little organs of pockets of white blood cells. And that's what they do, is they store white blood cells so that these lymphatic vessels are going to pull fluid out of the tissues and run that fluid into an organ that's carrying the white blood cell to filter out that dead tissue or whatever bacteria or viruses that are in there. Does that help?
Yeah, do they have any like reason why they are where they are?
That is a good question. I'm not really 100% sure. Yeah, I don't know, evolutionarily something, but yeah, cool. Okay, so our T cells. So we're gonna learn about T helper cells. So I know that this is confusing sometimes to call them CD4, but this is what they're called. They're called CD4 T cells. And I think I showed you guys the receptor thing, right? There's so many receptors on different cells. And all those receptors, usually that's how we can identify a cell, is by its receptor. So these receptors, these CD4 receptors, are specific to this type of cell that has this job. So somebody somewhere, I don't even remember what CD stands for, but they're called CD4 receptors, and these CD4 receptors are on these cells that have the role of a T helper. And then another type of T cell that is going to, so from the progenitor T, will make CD4 T cells and also make CD8 T cells. So CD8 T cells, even though they sound similar, they have a very, very, very different role, totally different cell. Yeah?
There's no information underneath this 7th degree and the 2nd degree stuff.
Oh yeah, I'll go over all that. Yeah. Okay. So a CD4 T cell is a T helper cell. A CD8 cell, a CD8 is just a receptor. It's how we identify these cells are a CD8, and these are called cytotoxic T cells, and that should already tell you kind of what their role is. They're cytotoxic. They kill cells. And we'll talk about how they do that. And then there's Tregs, which we're not going to talk about very much, but there's T regulatory cells. Sometimes they're called Tregs, and you'll see that in the literature. And Tregs, what they do is they kill T cells that are self-reactive. So cells that accidentally recognize a self cell, they'll kill that cell. So there are Tregs are like the police of the T cells, that they go, no, that's not a foreign antigen, that's a self antigen. So that they will kill that cell that's autoreactive. Very cool. they'll kill B cells too. Tregs will kill B cells also that are autoreactive. Okay, so let's go through them. So we talked about maturation. Remember, they are born in the bone marrow. Yeah.
Can we go back to the slide before this for a second? It
No.
says 5 types, but I only see 4 listed.
Oh, it's more than 5 types. There's tons of types.
okay, but we've
Tons
only
of
got
types. We're
4
only gonna
or
talk about
3 that we
these.
want to memorize.
Yeah, yes.
Okay,
Mhm.
thank you.
Yes, and I like this picture a lot because it shows you, this is the phospholipid bilayer of a T cell. Here's a T cell receptor. This is just a generic T cell receptor, so depending on what this TCR does. Then here's a CD4 receptor, here's an MHC1, of course it has a MHC1, right? So this is how we label and identify what type of cell this is. Okay, so T cell maturation. So we're not gonna go into the steps of maturation because I don't think they're important at this level, you guys don't really care, but basically what the point of maturation is, why a cell is born in an immature way. So remember I told you the story about the mom who had leukemia and her blood came out white. Those are immature cells, they're not effector cells. So there's maturity, they have a checkpoint, and that checkpoint, the first checkpoint is maturity. So they have to all the way grow up. They can't be immature. An immature cell is a non-effective cell. So once it matures, now it's called an effector, and once it's activated, now it can go and perform its role. So the maturity thing, the reason why that's important is one, because it's a checkpoint to make sure that those cells are doing what they're supposed to be doing, so it's one more checkpoint. But also, like for example, with leukemia, they'll never reach that maturity level, so they never will become an effector cell. When the hematopoietic stem cell is broken, it's gonna give birth to cells that will never mature. So those cells are actually ineffective and they won't go all the way to the maturation process. So yeah, it's just a whole nother step to it. But maturity, the only thing that we care about is there's multiple steps for a cell to mature, and the reason that our bodies have another way to check on this cell is because we don't want autoreactive cells. We want another checkpoint to make sure that they're gonna be doing what they're doing and they're not gonna kill us. Yeah.
I'm wondering if autoimmune issues have to do with problems with regulatory T cells, like if those aren't working.
It definitely is a Treg problem. It is also a Treg problem, but also I'll show you what happened. But it has to be a Treg problem as well. Yeah, and people that have problems with their Tregs have
Autoimmune.
Yes. Yeah, great. Okay, so here's our 3 types of cells: CD4, CD8, and Tregs. So our T helper cells are CD4s. So they help all the other cells. Now we know what they do, they're helpers, and I'll go through exactly what they do. But what they're gonna do is they turn on other cells of the immune system. So here they help activate other lymphocytes. Oh, I'm sorry, I shouldn't make that clear. These are lymphocytes. So we talked about leukocytes. These are also leukocytes. I hate to confuse you in that way, but what was your analogy you said? That all. Oh yeah.
All professionals are people, but not all people are professionals.
Yes, that was so good. So all professionals are people, but all people are not professionals. So all of these are leukocytes, but not all leukocytes are lymphocytes. These are lymphocytes. Yeah.
Is there a word for the leukocytes that are not lymphocytes?
No, we just call those leukocytes. Yes, yeah, that would be convenient. Okay, so these are lymphocytes, and they're in the lymphoid line. So remember from the hematopoietic stem cells, there's a myeloid line and a lymphoid line. These are in the lymphoid line. I can bring up that slide too. Yeah.
These are going to be all adaptive.
These are all in the adaptive. Yes, we're only talking about adaptive today. Great question.
So we don't call them myelocytes?
No.
Okay.
That would be helpful also. So here's the myeloid, here's the lymphoid progenitor, here's all of the myeloid lineage cells, here's the lymphoid lineage cells. So natural killer cells, I'll address it a little bit, but I'm gonna kind of like leave those because they are kind of ride the line between innate and adaptive. So I'll just kind of forget those. We can talk about them if we have time. Okay, so T helper cells. So they help activate other lymphocytes, and I'll talk more directly about what they do. They also release a lot of cytokines that help to attract other white blood cells, help turn on other white blood cells. So they are like the main helper. So if you were a virus such as HIV, this would be your target of choice, right? If you were going to target any cell that was the most effective, this is the cell that you would target because they're the most effective at helping all of the other cells. They call in other cells, they will turn on many other lymphocytes, but we'll go through a couple of those that they can turn on. Okay, so we'll go through these more specifically again and again. So our CD8 or cytotoxic T cells, they'll kill infected cells. So these are cytotoxic. So these aren't bacteriotoxic. They're not viral toxic. They're cell toxic. They're gonna kill the cell itself. So if the cell is infected with a virus or the cell has cancer, it's gonna kill that cell. So what they do is they are, I don't know if I wanna call them granulocytes, but they release, oh no, I'm gonna say degranulate though. So I will call them a granulocyte. We'll degranulate. These toxic enzymes called perforins and granzymes. And what perforins and granzymes do is they're toxic molecules, proteins that will break up that cell and kill that cell. So you want to double check these ones, right? These ones, you have to make sure that these ones are turned on and turned off and they're doing the right thing, that they're attracted only to cells that are infected, because you don't want cells going around killing things. These are highly toxic and they will kill cells, and if they are autoreactive, there's a big problem. And then Tregs, so what Tregs are is they will lower the immune response, so they release like anti-cytokines kind of, so they'll release cytokines that turn down the immune system. Also they'll kill autoreactive white blood cells. Okay, so let's talk about activation. So activation is kind of a tricky process, but the reason I dug into it, and the reason I wanted you to understand it is because I want you to understand what the point of the adaptive immune system is. Yeah, go ahead. Yeah, just that Tregs kill autoreactive cells. Yeah. Okay, I wanted to wait to the end. Should I wait to the end? Should I not wait to the end? I'll wait to the end. Okay, so I want to emphasize activation because this is why the adaptive immune system takes time. This is why the adaptive immune system is so powerful, because it needs this second step and it takes time to target the right target. So here when I say primary and secondary, that's what I mean by 1 and 2. This is a primary response. That means the first time that we're exposed. And here is the secondary response, the second time we're exposed. So remember I told you about Kennedy. I've told you that story a few times about how with the peanut butter. So the first time she had a reaction, she probably had a mild reaction, and maybe even the second time, maybe even the third time. The fourth time it was so reactive that it's deadly. And so the primary response, usually is a slower response. And this is why we feel sick. This is why we get sick, because this hasn't kicked in yet. So this primary response is the first time that we're exposed to the pathogen or a vaccine, right? Because that's the first time we've been exposed to it is when we get the vaccine. Okay, so what's gonna happen is to turn on a CD4 T cell, this is our most important job, is to turn on that CD4 T cell. We have to be able to turn that one on. And how we do that is through dendritic cells. So remember, dendritic cells are the innate cells that will antigen present to a CD4 T cell. So this dendritic cell is going to antigen present on an MHC2, and there'll be cytokines in the area. We'll release some cytokines. There might be cytokines from other cells or whatever it is to be able to turn on that CD4 T cell. Now the CD4 T cell is turned on and it can do its job. So now it'll go and help other leukocytes or lymphocytes. And there is a memory T cell, which we're not going to talk very much about memory T cells, but there is a memory T cell that will be stored forever. Now the CD4, whatever antigen was presented on this MHC2, those CD4 T cells will remember forever. We'll talk more about memory. But the second time that we're exposed to that infection, now even cytokines can trigger those CD4 T cells. The cytokines that triggered them the first time, those cytokines can trigger them the second time. So now if they see things like, they're called interleukins, if they see these cytokines, then it will trigger the CD4 T cell to turn on and go start helping. So CD4 T cells, they're not as regulated as some of the other cells because basically their job is to go and help them turn on other cells. So a dendritic cell will present an antigen on an MHC2 and it can be turned on really easily the second time around. Okay, so that's activation of CD4 T cells. In your book, it does a good job of showing you little pictures. I like the little pictures it shows you in the book and it kind of takes you through. This is a CD4 T cell. This is a CD8, but I left it in here because here's the T helper. The T helper is going to help the CD8, which we're going to learn about next. Okay, so a CD8 cell, even though they sound similar, they're very, very different. So an activated, the first step on primary infection, the first time that we're exposed, a CD4 T cell has to give a signal to the CD8 cell to turn on. So if your patient has HIV, for example, and has low CD4 numbers, then CD8 cells will not be turned on. You have to have those CD4 T cells to be able to turn on a CD8 cell. So a dendritic cell, so this is where dendritic cells get funky, and this is where I want you to clarify and ask questions and make sure that you understand this part. So dendritic cells are crazy. Dendritic cells are actually really, really cool. What they do is they can present on an MHC2. Remember, they're professional antigen presenting cells. They present really fancy on that MHC2, and that's their main role. But this is something funky and cool that they can do. They can present an antigen on an MHC1, which would tell it, kill me, right? That's usually what the CD8 cell is looking for, a presented antigen on an MHC1, because that means the cell is infected. Is everybody with me? Do you want me to go over that one more time? So remember MHC1 and MHC2. So I'll draw a human cell. This is MHC1. And then I'll draw a let's draw a dendritic cell has a lot of these things called dendrites. They look like pseudopods, kind of. And this is an MHC2. Okay, so MHC1 is all nucleated cells. MHC2 are only the fancy antigen presenters. Dendritic cells, macrophages, B cells. Okay, so what a dendritic cell can do, so this one, if there's an antigen on here, there's a virus in here, and there's an antigen on here, like a spike protein. And now it's saying to the CD8 T cell, here's a CD8. This CD8 cell turns on and it's gonna release perforins and granzymes to kill this cell because it recognizes a foreign antigen on an MHC1. You with me so far? Yeah.
So you just call it like a self-destruct button?
Yeah, it's like, it's kind of like self-destruct, but it's more like, if you imagine like a pathogen has been set loose in the population, then there were cops that went around and put those people in jail. Hopefully they didn't kill them, or they killed the people that were carrying the pathogen so they wouldn't get anybody else sick. So these are the cops that come and kill this cell so that no other cells get sick. So that's the role of the CD8, perforins and granzymes. But this CD8 has many checkpoints that it needs to turn on because it can't go around killing things. The more it goes around killing things, the more in danger we are from this very, very powerful cell. Yeah.
So would the CD8 be a natural killer cell?
A CD8, no, natural killers are different, but they work in similar ways.
So
Yes.
do natural killer cells release those two enzymes?
Release the same enzymes,
Okay.
yes, yeah. Okay, so CD8 cell. Okay, so here's our dendritic cell, and in this dendritic cell, we have an MHC2. And that MHC2 can antigen present something that it phagocytosed, like the virus or the bacteria or something like that. It phagocytosed it, it can present on MHC2, which that's going to tell it to sound the alarms. So this is the role of the MHC2, is to call in other cells, to turn on other cells, to present to other cells, to tell everybody what's going on in the body. But also it has this other really cool thing that it can do, and that's unique, that it can present on an MHC1, but not say kill me. And dendritic cells are the only ones that can do this. And they're going to present on an MHC1 because this is going to tell the CD8, it's another checkpoint. It's just another way to turn on this CD8. The dendritic cell presents the antigen on an MHC1 to the CD8, and that is telling the CD8 that there's a self-infection, but it's not gonna kill this cell. It's only gonna kill this cell with the same antigen. Yeah.
Well, why would you need to have MHC1s do it if isn't that what the MHC2 does already?
So the
Like
MHC,
if the CD8 sees the MHC2, isn't that turn the CD8 on also?
so the CD8 does see an MHC2? Does it see an MHC2? I don't think the CD8 sees an MHC2.
Oh, okay.
Yeah, yes. So the CD8 sees MHC1s
Okay, so it just has a receptor for the one.
for the one. Yeah.
So the antigen on the MHC2 of the dendritic cell, the one on the right, like the red dot, you have, like in the receptor,
Yeah.
that's coming from extracellular environment, correct? That's not coming from inside the dendritic cell? Because MHC2s present stuff from the outside of the cell, right?
Yes, but it phagocytosed it. So it
Okay.
phagocytosed
Got
it and then it chewed it up and spit it out.
Okay, I see.
Yes. So the way that antigen presenting cells can phagocytose is because they keep it in a special little pocket. So it's not gonna kill this cell. All of our cells don't have the ability to do that. So only these cells have the ability to actually be infected without being infected.
Okay, and then the antigen on the MHC1 there, is that also phagocytosed out?
Yes.
And then presented there?
Mhm.
To activate the CD8?
Yes.
And how is it gonna kill the other cell?
How does the CD8 cell know to kill this cell and not this cell?
Well,
Well,
why
when-
is the antigen on the dendritic cell? How is it?
it presents on a different, so here is a different receptor,
Okay.
and this receptor is going to meet up with this receptor, and then that receptor is just going to tell this to turn on. Go.
So it would have to see the dendritic cell's MHC1 first before it saw the MHC1 on a regular cell.
Yeah, I don't know, I think it can be done simultaneously, I think that they can meet up together
Okay.
at the same time.
Like a proximity thing.
Yes, yeah,
Was the point of the MHC1 being able to present on the dendritic cell, is that part of the adaptability of the adaptation?
so what,
Is that how it
so,
works?
so the adaptive part of it is, what the adaptive means in the adaptive immune system is that this cell has to be turned on with different things. So what adaptive means is, that this cell has to be turned on. That it can't just go like mast cells, when it meets up with an antigen, it'll just degranulate. Neutrophils, it'll just degranulate. That they don't have to go through this other checkpoint of CD4 activation plus dendritic cell activation, plus it has to meet up with the antigen on the cell, because, so it has checkpoints for it to become active. But also the specificity of, which I'll talk to you more about specificity, but this antigen has to match this antigen exactly. And
So
then
is
it's
that
saying,
why it's still going through all the checkpoints when you get infected the second time? It's just being able to do it like a lot quicker.
yeah, so I'll go through what happens in the second time. Yeah, no, that makes sense. Yeah.
So on the board underneath first and second degree, that's just activation conditions, essentially.
Yeah, so once it sees it again, let's go through that right now, that's a good point, yeah.
Do you know any good videos that illustrate this?
Yes, yeah, I can post some videos, that's a good,
That'd be
yeah,
great.
I'll post some videos. And I'll filter out some videos for you, just because it's extremely complicated, and I'm trying to make it step 1, step 2, step 3, like I'll try and find some simpler ones if I can. Okay, so MHC1 presents an antigen, MHC1 presents an antigen to turn on CD8, but also don't forget you have to have a CD4 has to be turned on, and to be able to turn that CD8 on. So it has here CD4 help, dendritic cell with the MHC1, an infected cell with an MHC1, plus it needs cytokines to get it going. So that's
So
a
is it
lot of
like
checkpoints.
At the bank when you need 2 keys to get the thing open?
Yes, double check safety, 100%, yes. So the second time that this specific CD8 cell is going to run into another cell that's infected, now it doesn't need all those checkpoints, it's already on. So now this CD8 cell, and actually this CD8 cell can do something called cloning, so it can clone itself, because this exact key matches this exact lock. So now that CD8 cell can clone itself, but also that CD8 cell will find another antigen. So it killed this cell. Here's another MHC1, because the cell next to it probably has an infection, and the cell next to that one probably has an infection, because the virions released, and those cells are probably all infected in the area. So here is the MHC1 of the cell next to it, and it doesn't have to be turned on anymore. Now it can just release its perforins and granzymes. So that's it. It builds, it becomes more and more effective. That's why it can clear our body. But also these can be stored forever. So now they don't have to be turned on anymore. Anytime a CD8 will find this exact antigen for the rest of your life, it will meet up and kill that cell, and it never has to be turned on again. Yeah.
So are you saying that 3 key with the cytokines, the MHC1 and the CD4 is only a one-time thing? That is the
first
activation.
exposure.
Okay.
Yes. Yeah.
For CD8, and
great question. Only one type. Only
So-
one PAMP. Only one.
Okay, so
Yeah.
if one CD8 cell is like, hey, I like this antigen, I'm gonna choose to attack this for the rest of my life.
Yes.
It can't have more than one.
Nope, can't have more than one.
Perfect.
Yes, yes, great question. Yeah, and we'll go into why. Okay, so the next time that your patient has an infection, or the next time that cell will see the infection, now those cells can be stored forever in the lymph nodes and in the secondary lymph, all the organs, your lymphatic organs. Now, if you get that virus again, you won't even notice it because that it will kill that cell immediately. And so sometimes even with like virally infected cells, cell death doesn't really feel that good. So sometimes you might feel run down, you might feel like meh, but generally it will kill it. It will kill those cells, and the other cells will clear it because they're all automatically turned off. Yeah.
So when it recognizes it again, it's due to the memory cell?
Yes, because that cell right here can clone itself. So here it can go over and kill the next cell, but also it's going to go through a process called cloning. And so now this specific CD8 cell that matches this specific antigen will be stored, and those are called memory, actually. They're called memory T cells that are stored forever now.
But they're just identical cells to the first activated CD8 cell?
Yes, so that match, this receptor matches exactly this antigen.
Like, there's nothing like special about them that has anything different than the first one?
Yeah, no, they're
Okay.
just the only thing special about them is that this lock matches that exact key. Yes, awesome. Yeah.
Sorry, one more question.
No, please.
So the T regulatory cell is regulating both the CD4 and CD8 and turning them off and on?
So what the Tregs can do is they secrete cytokines that will block these cytokines. So if there's a lot of cytokines that trigger the Tregs to start working, the Tregs can start blocking some of these enzymes and some of those cytokines so that it will lower the immune system. But also, if this is not a virally infected cell and this cell is not broken, and there's something presented on this MHC1, something happened, or maybe this CD8 receptor accidentally was born and accidentally will bind to anything on the cell at all, actually, or anything in the environment, and it starts releasing perforins and granzymes that's attached to that cell, Tregs can kill that cell.
Oh, so they can completely kill.
They can
CD8
kill the
T4 cell.
CD8 T cells that
Okay.
are autoreactive. Yeah.
How much should we know about perforins and granzymes at this stage?
That's it. Just perforins and granzymes kill cells.
Okay.
Yeah.
Can you very briefly talk about again how the CD4 activates the CD8, like just right before this?
Yes. So CD4s have different roles. So some CD4 cells will actually bind to cells to turn them on, and some can just release cytokines to turn them on. So just know CD4s are helpers. They either release cytokines to turn on a cell, or they'll bind directly to a cell to turn it on. So they kind of have different roles depending on what we're talking about. But CD4s turn on other cells. Okay. So that's CD8 T cells. Those are probably the most complex. Well, but they're complex because there's so many steps. And there's so many steps because we don't want them killing things randomly. We want to make sure there's a checkpoint. Okay. So here is a thing from your book that helps T cells, CD8. Now you have a memory CD8 T cell activated, perforins, granzymes will kill the infected cell. Your book does a good job explaining these. If you go through the book on these sections, it really does help. Okay, so what is a B cell? So let's move on to our Bs. So we start with an effector B cell. Those effector B cells, those are kind of like progenitor cells. Try not to get mixed up into the lineage situation, but a B cell is like a T cell is a progenitor cell that can give birth to other types of B cells. So we're just moving down the line, down the line, down the line. I know it seems crazy, but like when you think about, for example, I've given you guys this example one other time, but this is a terrible pen. Like totipotent stem cells give rise to every single cell in our body. That's why people use them in like in a lab setting to build other types of cells, neurons, skin cells, chondrocytes, any kind of cell you can think of, muscle cells. So those totipotent cells are super potent. They can make any cell type. But like if you say like a B cell is multipotent, so a B cell has a function, but also it can give rise to other types of B cells. So my example I was going to give you, I'll just throw it out there, but like the basal cells of your skin will give rise to more basal cells and give rise to keratinocytes. So they'll make more basal cells and they'll make keratinocytes. B cells will give rise to more B cells and give rise to other types of cells. So, okay, so B cells are found throughout the body. They're born in the bone marrow. They are mature in the bone marrow as well. They're very versatile. They can do all kinds of cool stuff. And they make these two types that we're going to talk about. So the effector B is this B cell that floats around, and it can give rise to two other kinds of B cells. One is called a plasma B cell, and one is called a memory B cell. The point of B cells are to make antibodies. That's the whole point. Antibodies are a huge part of our adaptive immune system. We have to have antibodies, and antibodies have a really cool role. So here, if you look on the phospholipid bilayer, here's MHC2. Remember, they are professional antigen presenters. Here's MHC1, which they all have, and here is an antibody. So antibodies are funky because so also do not forget that. Antibodies are also called immunoglobulins. Antibody, immunoglobulin, antibody, they mean the exact same thing. So here is the antibody / immunoglobulin that's attached to the B cell as a B cell receptor. But what B cells are able to do is spit those out and let them float around in the environment. So it's like having a receptor on the surface, but also it can get rid of the receptor, which is crazy. Okay, so let's talk more about B cells. So B cell maturation happens in the bone marrow. They mature and become a mature B cell when they're in the bone marrow. Yeah.
So would you say that they're being stored in the bone marrow or no?
They're stored in the bone marrow too, yeah.
Okay.
Yeah, okay, so we're gonna talk about B cell maturation. So B cell maturation is a really funky, cool thing, and this recombination is the reason why I wanted to bring this up, is because it's the way that we can find one antigen and match it exactly, and the reason that this antigen binds to this receptor is not just magic. That they have to have a chemical match. There is a chemical lock and key. So if you imagine what that receptor looks like on a cell, and there's stuff floating by, not everything can turn it on. Only specific things can turn it on. So everything that's floating by, it has to have an exact lock to the key. So that exact lock to the key is crazy because of all of the things that we inhale and that we eat and that we're exposed to every day, all day and night, all the time. There has to be a lock for every single key that could make us sick. And literally there's trillions of them. That's not an over-exaggeration. There's trillions of locks that can find different keys, of possibilities, of being able to find every key there is out there. That's how powerful our immune system is. So what happens is when a B cell is maturing, the DNA changes. So this is when we talked about DNA. The DNA is the same in your eyes, in your lungs, in your liver, in your skin, and we have all of the same DNA in every single cell. There's only a couple of exceptions. One exception are the gametes, the eggs and sperm. Eggs and sperm have half the number of chromosomes, so they don't have an exact match as the whole body. Another exception are the red blood cells. Red blood cells get rid of their nucleus, so they don't have a nucleus at all. Another example are B cells and T cells. B cells more specifically. So B cells, what the chromosome will do is actually the chromosome cuts. There's actually cuts in the chromosome itself that it's going to cut and rematch so that it gives a different protein profile between every single cell that's born. So every single B cell, every single antibody right here, let me show you. Every single antibody from one B cell has one antibody type, and that one antibody type is unique to the trillions compared to every other B cell that's born. So what happens to this antibody is the chromosomes that the DNA that makes the RNA that makes the proteins, the DNA is different in every single B cell that's born. And the reason it's different is because of this thing that's called VDJ recombination. So there's a segment called V, there's a segment called D, there's a segment called J, and what happens is the actual DNA itself is cut into different varieties, and that will give a different variety of amino acids on these variable regions of an antibody. So let me draw a picture of what that looks like. So an antibody, remember, is just a B cell receptor. So if we can draw the B cell, the antibody, obviously it's not to scale, this is a huge receptor. Looks like this. So draw that on your paper. Everybody draw that. So this is a B cell. This is an antibody or an immunoglobulin. This section right here, this little piece is called a light chain. This piece is called a heavy chain. So there's 2 heavy chains, 2 light chains. This piece out here is called the variable region. It's also called an FAB. I forget what that stands for. It's like functional something. That's called the FAB region. This region right here, this whole region here, is called the constant region. Or it's called FC. So, but there's 5 types of constant regions, and there's trillions of variable regions, because these amino acid sequences, there's trillions of different possibilities of amino acid sequences because of the DNA is cut and spliced for the region of the chromosome that codes for this area of the antibody. Did that make sense? So there's so many variations on here because what they're trying to do is try to make a million different locks, trillions of different locks for the type of key. So what type of key is this? What type of bacteria is that? What type of virus is that? What type of pollen, dust mites, chemicals, any single thing that we're exposed to, there has to be a variable region to match it, to get rid of it. Anything foreign that gets in our body has to be able to take care of that. So this is what an antibody is and what it looks like. The reason for that variation is because of a change in the genome. So if you look at the genome of a B cell, it's going to be different than the genome of a skin cell because of this little region is cut up. So here's a picture of the antibody of a immunoglobulin. And then I'll go through what these cells can do. Maybe I'll spend a couple more minutes. So an effector B cell, so the effector B cell, what it does is it can phagocytose, it can antigen present, and then also it can do this funky thing where it can differentiate into different types of B cells. So it's an effector cell itself, but also it can give rise to other types of cells. So it can differentiate to different types of cells. So these two types. So the way on the primary exposure, when we're first exposed, it can phagocytose whatever that is, but also it needs CD4 activation. So here are important CD4s again. It needs some cytokines to turn it on, but then also that VDJ recombination, do I want to say that there? Hold on, let me think if that's what I want to say. I don't want to say that.
What is VDJ?
I want to say it has to go through a cloning process is what I want to say. So remember, so here, there's a V region, a D region, and a J region. The V region, so say we have 46 chromosomes, 46. On one chromosome, well, it would be on two, because it's the mother and the father. There's a region called a V, a D, and a J. And this area of the chromosomes of the B cell, here's the B cell nucleus, these chromosomes code for this area of the antibody, for the variable region of the antibody. Maybe let me clean that up a little bit. So here is the variable region, and here's the constant region. So this variable region right here has so trillions of different options because these are cut in different places and spliced. Basically, it's like splicing, so that it can have a AATC or an AAC or a CCAA or a CA. It's cut out different parts so that there's a different amino acid structure in these variable regions. So this happens during B cell maturity. So what happens is this one B cell is going to be turned on because it met its match. It phagocytosed something, and now this B cell is turned on. Now it's going to clone. So this is like a fingerprint. It's like a unique VDJ recombination that found its match. So now that it found its match, it has to clone and make thousands of all B cells of the same match, of that same antigen that it phagocytosed. So it found this antigen, this is an exact lock for the exact key, it phagocytosed it, now it's gonna clone. Now it's gonna make all these other Bs, but also what it's gonna do is it's going to differentiate into plasma cells and memory B cells. So it can make more effector Bs, it can make more memory Bs. So now that it found its exact match, all of these are going to match this exact antigen, this exact antibody antigen match. So a different B cell that has a different antigen doesn't even recognize this, has no idea what that is. It doesn't recognize that. It's only this one that recognized this one that's going to be able to clone. And doesn't that make sense? Because now say that's poliovirus. Now that B cell recognized a spike protein from that poliovirus. Now every single one of these matches poliovirus. So now those can be stored forever, they can be effective against the polio, and they're specific to that polio. So now they have to clone. Now the second time that your patient is exposed to polio, they'll phagocytose the polio. They need CD4 help again, even though this is the second time. Then they go through, then they need cytokines as well, but they don't have to clone. Now there's already hundreds of B cells that match this exact one. All they have to do is be turned on. That's it. But now that we already have tons and tons of B cells. But there's one more cool thing that they do. We'll figure out what plasma cells are. So let's go to plasma cells. So plasma cells secrete antibodies. Sorry, I'll be 2 more seconds. So this just talks about B cell cloning. You can look at that. So this is what plasma cells do, is they secrete antibodies by the hundreds of thousands. So an antibody can be docked as a B cell receptor. So this antibody can sit on this B cell forever, but also this antibody can be spit out, and this antibody matches exactly the antigen. So matches that exact polio. Now you have these proteins floating around in the body that match polio forever. So now it's these proteins. So what do these proteins do? So I'll just introduce what these proteins do really quickly, and then we'll talk about this again on Thursday. So antibodies are incredibly powerful because now you have proteins floating around that recognize exactly polio and what it is. So what antibodies can do that have been spit out by what are called plasma B cells. So this one differentiated into a plasma B. This plasma B cell is now going to spit out antibodies by the thousands, tens, hundreds of thousands. So what antibodies do are these 4 things. So they can opsonize, which is called tagging. So now what they're doing is that bacteria is no longer able to function anymore. And they tagged it so that the macrophages and dendritic cells can go and swallow it, and neutrophils can go and swallow it. So they can tag it, they can agglutinize. So agglutinize is gluing. They can glue those pathogens together so that they're no longer effective and they can't go and spread their endotoxins or exotoxins around. They can activate. Remember we talked about the complement proteins. So there's the classical pathway and the, what is it called? Classical and, I always forget the other one. Alternate. And the alternate pathway, but in the classical pathway has to have an antibody already stuck to it. And that antibody that's already stuck to it, now the first complement protein can attack and form the holes. Also they can just neutralize, so they can surround it so much so that it doesn't function anymore. And it can't get its nutrients, it can't attach to the lining of the cell wall or whatever it is. So that's how strong antibodies are, and they are specific for this type of bacteria because they've seen it so many times. So for example, the person next to you has a cold. Well, your body already has antibodies that are running around all of your whole bloodstream that attack that virus. So you won't even notice you're sick at all. The virus is like just floating around doing its thing and you can't even tell that it's there. So that's how potent antibodies are. They're specific, and that is why we have to have a primary and secondary response, and why that secondary response is so important is because already all these B cells are ready to go. And they're already specific to that one type, and now they can attack without having to be activated again. Does that make sense? You with me? Okay, so let's finish there and we'll go, we'll keep going next time. We're almost done. So what I'm gonna do for the quiz for next time is for our quiz for next week, for next Tuesday, we're going to do all immune systems. We're gonna do innate and adaptive all together in the next quiz. So review your innate again, go through some of those questions that were tricky, go through all of that innate again. We're gonna do innate and adaptive together, even though on Thursday we're gonna start talking about diagnostics. I won't ask you about diagnostics on the next quiz. We'll keep that for the next quiz after that. Yeah.
Last week, I think we made it through like half of the, innate immune system, like the lecture slides.
Mhm.
Are we gonna go through those again, or should we worry about those?
We didn't make it all the way through.
I feel like we only made it halfway through.
Oh.
Yeah, there was some.
It was like 80 something slides, but we only got halfway through it.
Oh, okay. I will go, let me look and see what we got through.
There's a red slide in your innate slides, and you stop there.
Oh, that's it then. It's just sometimes I put in extra slides to like stuff from like previous semesters or I
okay.
forgot to delete.
It was like
So
So
everything
put it
past
off.
the red we can just throw away.
Anything past the red, throw away. Yes, yes. Usually I delete them for you, but sometimes I keep extra slides at the end if like there's something important that I want to put back in there. Those are like my bonus slides.
Are all of B cells, so for the B cells, the DNA is, I'm not giving you, so the DNA is.
B cells are
Oh, they are, aren't they, what's the name of the professional?
all cells have nucleus. Except for red blood cells.
Oh, we talking, okay, so like non-nucleated, so MHC1 is all
I'm sorry, what?
MHC1.
Every cell has a nucleus. The only ones that don't have a